Welcome to now our first open journal club session of ComputAge community. What is ComputAge community? It's a kind of DeSci organization dedicated to building an ecosystem of computational biologists who are interested in longevity. And today, I'm happy to introduce to you Evgeniy Efimov, who is a great aging researcher and an expert in the different topics of omics data analysis, especially DNA methylation. He will tell you about his understanding of, I think, very important today, very important model of called ground zero model of embryogenesis. If you don't know about ground zero model, don't worry. Evgeniy will give you a short background, explain basic terms needed for understanding. And our agenda today is 35, maximum 40 minutes for speech of Evgeniy, and then 40 minutes of discussion with questions. Please ask questions after the discussion, because we want to be in our timeline. So, without further ado, Evgeniy, please start.
Okay, can I please be allowed to share my screen?
Sure.
Okay, just a second. Can you see it?
I can.
Yeah, it's okay.
Okay, great. So, yeah, hello everyone again. My name is Evgeniy Efimov. Thank you, Dmitry.
I'm sorry, Evgeniy, we see a text.
Text.
Yeah, some window with text to slides.
Right now? No?
It's seen. Yes, now it's not. Yes, only the slide.
Okay. So yeah, thank you, Dmitry, for introducing me. Today, I'm going to discuss the ground zero phenomenon, or in other words, how nature rejuvenates every one of us during the process of embryonic development. A small disclaimer, I'm not sure how expert you are all in these topics I'm going to discuss, so something of it you may know already, but I hope there will be something new for everyone as well. Everything I'm going to discuss right now is basically the compilation of multiple articles. So let's start. Over the past few decades, it has become evident that cells can be reduced artificially in the process of cellular reprogramming by applying some transcription factors or chemicals, such as, for example, the Yamanaka factors. But in fact, nature has already been rejuvenating animals and basically every other organism for a very long time during the process of development. For example, if during the process of embryogenesis in animals, cells from two quite aged organisms' parents fuse together to give rise to a completely new young organism. And then there must be some rejuvenation event somewhere along the way, which would correspond to the point of minimal organismal age. By the way, can you see my pointer? Yeah, okay. So yeah, this this rejuvenation event would correspond to the point of minimal organismal age, after which the organism would start aging. And this event likely happens after conception, somewhere after conception, and somewhere before birth. Yeah. So, but how and when exactly it happens, it occurs, it's still quite a mystery. It's still very much unresolved. And knowing the answers to these two questions would benefit a lot to the cellular reprogramming and rejuvenation therapies. But before we go deeper into the rejuvenation, let me first remind you a little bit about embryology overall, the fate of every animal on this planet. Everything starts with the two parental gametes. When a sperm cell and an oocyte fuse together, the sperm fertilizes the oocyte, and then they proceed through a very, very nice, great and beautiful period of embryogenesis, after which a birth happens, and after birth, every animal undergoes some kinds of larval stages or childhood, and then we all go through metamorphosis, well, to some degree, after which animals are fully sexually matured, and they can meet each other, and make the cycle repeat itself again. Here, I'd like to pinpoint this red arc I've drawn, which is the period that interests us the most, because we assume that during this exact period, some kind of rejuvenation must occur. I've pinpointed the gametogenesis in kind of another color, because as we understand, the germ cells, such as sperm and oocyte, do not appear to age as much as the other somatic cells. But still, they must accumulate some kinds of damage over the course of organismal life. Therefore, this damage must be somehow eliminated during this embryogenesis process, so that the new organism would not be even a little bit aged compared to the completely young organism. And yeah, a little bit of embryogenesis. Embryogenesis itself can be broken down, as you can see, into three main stages: the cleavage, the fertilization, of course, the cleavage, during which this first single oocyte breaks down into many small cells, the gastrulation stage, during which the main embryonic layers appear, such as ectoderm, mesoderm, and endoderm. And then there is this organogenesis, during which all of the complex tissues and organ systems, etc., appear and are ready to produce a new organism. A little bit about the early stages of embryogenesis. Here you can see the timeline of it, from fertilization, basically, to the formation of initial embryonic layers. Basically, the outer layer is called trophectoderm, and it will become what is implanted into the mother and what connects the embryo to the mother's blood flow and tissues. And there is also this inner cell mass, which differentiates into the thin layer of hypoblast, which will also become extraembryonic, temporary extraembryonic tissues, and the epiblast. The epiblast alone gives rise to all of the beautiful organisms that you and I present to this world, because only these epiblast cells will then differentiate into embryonic tissues and then make up the whole organism. And as you can see, the timeline for human embryos and for mouse embryos is a little bit different. Humans develop a little slower, but the general idea stays the same. And after gastrulation, when all of the three main embryonic layers, such as ectoderm, mesoderm, and endoderm, are established, they quite rapidly differentiate into so many different complex systems, tissues, organs, etc. And yeah, this is just beautiful how we all develop from just some small identical stuff. Yeah, and I also wanted to note here that in embryology, cells are usually notated as half days. For example, embryonic day 9.5 or 11.5, etc. It's a really common notation, so don't get intimidated by it. It's just embryonic days since conception. And here, once again, I only wanted to introduce you to the complexity of this beautiful process, because this picture shows how different tissues develop in mouse embryos over time. This line shows development over time and through space. This is the cross sections of embryos at every temporal stage. And the tissues are painted with RNA sequencing data. I just love how enormous this amount of data is, and at the same time how really difficult it is to get any biological sense from all of these pictures, because when we look at them, well, how can we guess at which point we have an age 0 anyway? So yeah, just looking at the pictures and just reading RNA sequencing is not a good idea to approach this age 0 aging problem. Another instance of complexity. This graph shows the whole tree of embryonic pathways through which all cells differentiate, starting from this one little point of moral cells here in the beginning, when all cells are completely identical to each other, or almost completely identical, and ending somewhere a little before birth. The days are shown for mice, and well, most of the experiments I'm going to show were made for mouse embryos. Yeah, because there are some troubles when you want to analyze human embryos, by the way, but I'm not going to go into that today. So there are 3 main periods of lineage divergence, of differentiation periods. The first one is this little point here, this fork here. It's called plastulation, when morula differentiates into the outer layers, into the outer layer and the inner layer. Then we have this, well, quite wide process of gastrulation. Basically, it's this point here, when the epiblast differentiates into the 3 layers: ectoderm, mesoderm, and endoderm, again, reminding you all these terms. And then we also have this some moment during organogenesis, when lineage progenitors are cells which will give rise to some subpopulations of cells. For example, some will give rise to all the muscle tissue cells. Some of them will give rise to the immune cells, etc. So yeah, 3 major periods of differentiation we have. And my hypothesis, which is based on the review of multiple literature sources, is that the period of gastrulation, well, maybe a little bit before that and a little bit after that, is the period of is the best candidate for ground 0, for the age 0, because well, before gastrulation there are 2 key events that I'd like to mention here: the zygotic genome activation and totipotency. What is the zygotic genome activation? It's the event when, well, after conception, the zygote has to survive for a little while from the proteins and RNA that it inherits from the oocyte, because the zygote itself is born, is created completely silent. But over time, approximately by this morula stage, zygotic genome gets activated completely, and it no longer relies on the material inherited from the mother cell, from the oocyte. And then we have also the totipotency, which is this state of complete of any possibility. From this state, a cell can enter any fate. As you can see, this the cells here can give rise to any other cell types. So this is called totipotency. And also here I claim this name this pluripotency stage, which is basically somewhere here. These are the cells that will give rise to all the embryonic tissues, but they cannot make any of these orange points here of extra embryonic tissues. So why do I say that? Because before gastrulation ends this zygotic genome activation, and also before gastrulation, there is no need to rejuvenate extra embryonic tissues. They will not go into the new organism, therefore they needn't, the nature doesn't have to rejuvenate such tissues. So there is no real evolutionary pressure to rejuvenate these ones. But these cells that will become the future organism, there is some pressure. Yeah, so we could hypothesize that H0 begins along with totipotency, but also we have some other considerations. This totipotent embryo is substantially different from the pluripotent one, so different that we cannot, for example, the Yamanaka factors, as you may know, they reprogram somatic cells into the pluripotent cell. But it's really, really difficult to combine, to create such a mixture of transcription factors that would directly reprogram somatic cells, all of these here, into the totipotent state. And also, totipotent chromatin is really, really opened, extremely opened, which means that there are so many genes that can be transcribed. Basically, almost all genes can be transcribed at this point. And this is bad for the cells. Why? Because among many, well, good genes, there are also bad genes, such as transposable elements, which can break genes apart if they insert themselves into the genome. And there is this hypothesis, which surprisingly hasn't been quite proven yet, that if totipotent embryo gets cultured for quite a long period of time, then the actions of this junk DNA, such as transposable elements, will induce a lot of insertions, a lot of deleterious, deadly insertions into the genome. So, judging from all these considerations, I assume that totipotency is quite an unstable state, and that only the pluripotent period of embryogenesis from blastocyst. Well, here I show this pale red line, which is kind of the beginning of gastrulation. And through this whole period of gastrulation until the beginning of organogenesis, this period deserves credit, in my opinion, as the H0 or ground zero of organismal life. Yeah, so after this long introduction to embryogenesis, which I hope you got some idea of, at least, let me briefly describe the process of artificial reprogramming. I think it's extremely important to the matter we discuss here, because it has also been shown to rejuvenate old cells. So, as some of you may know, reprogramming is when we apply some transcription factors or chemicals onto the cells, and these transcription factors make cells erase all of their program over time and become completely pluripotent, so that they can create any type of cell. These completely reprogrammed cells are called induced pluripotent stem cells, iPSCs. And this process of reprogramming can be split into three major phases: initiation, maturation, and stabilization. The further a cell goes through all of these stages, the more it forgets its identity on average, and the more pluripotent it becomes. We can stop reprogramming at virtually any point by removing the transcription factors, such as these OSKM or KMS factors, and we get different results. For example, if we don't let cells complete this initiation phase, then we can induce the transdifferentiation process, during which a cell can change its program directly from, for example, a skin cell to a muscle cell or a neuron cell, etc. And if we stop at somewhere at maturation, then we can get the alternative stem cells. For example, neuronal stem cells, cardiomyocytes. These are the cells that can give rise only to a subpopulation of cells. For example, the neuronal stem cells will create only subtypes of neurons, and cardiomyocytes only the subtypes of muscle cells, etc. So these cells are not completely pluripotent because they cannot create virtually any type of cell. Only the ones which pass into this stabilization phase, only these cells can become pluripotent and create any other type. Coincidentally, not only cell fate, but also aging is reversed through reprogramming. And the deeper into pluripotency we go, the further along this hill we go, on average, the more rejuvenation occurs. For example, transdifferentiation doesn't appear to reverse age at all, and iPSCs seem to be as young in terms of some transcription profiles and epigenetic profiles. They appear as young as the embryonic stem cells, even if they come from really old people. So now comes the time to combine all these observations we've made previously into one meta-hypothesis. We know that during the maturation phase of reprogramming, cell identity is being lost, that the factors of pluripotency begin to work, and the cells reach such a state in which reaching full pluripotency becomes inevitable, well, by the end of maturation at least. Also, we know that aging factors are erased during this process, and that age 0 is reached again by the end of maturation. Judging by at least, first of all, by epigenetic clocks, which I'm going to talk about later, and by physiologically, I mean, phenotypically, they look, not phenotypically, morphologically, they look, well, and phenotypically, I suppose, as well, they look like young cells, completely young cells by the end of maturation. From the other hand, we know that during gastrulation of embryogenesis, pluripotency factors fade and lineage specifiers kick in, which means that pluripotency is being lost, and differentiation, the choice of some certain programs, becomes inevitable by the end of gastrulation. However, what we don't know is whether some aging factors begin to accumulate on this stage, during this stage, and whether age 0 is passed. So, merging these two conclusions, I suggest that maturation in reprogramming is reversed gastrulation, or vice versa. But the question we have now is whether the ground zero of embryogenesis happens during gastrulation. So having a hypothesis is, of course, a good thing. We can start at least from here, but can we now check it somehow? It's kind of difficult to track aging or rejuvenation in cells that undergo reprogramming or development, because the common markers of organismal aging, such as blood pressure or inflammatory markers, are not applicable here. You cannot measure blood pressure of cells. Luckily, the field of aging knows an instrument for it, and that is aging clocks. There are several variations of aging clocks, but today we will focus only on the epigenetic ones. Or, as you may know, epigenetics include modifications of DNA, shown on this slide as these black and white circles. Also, we have these histone modifications. Histones are the proteins which are bound at DNA. And we have also some chromatin stuff, openness or closedness. All of these markers are useful if we can build clocks from any of these markers. But when we talk about histone modifications, for example, the patterns are kind of how do you say it? Kind of established. For example, we know that some histone modifications are activating the genes beneath them, some are repressing, but with DNA methylation, the situation is not as evident, it's not as transparent, because in some cases, some positions in the genome can be demethylated or methylated, but it doesn't say us to tell us anything about if this position activates the gene nearby or represses it. But methylation does change with aging in a specific way. The main idea of these changes is that, on average, when cells are young, their methylation profiles are strictly bimodal, which means that for some methylation sites, most cells are either hypermethylated, either methylated or demethylated. But as we grow older, these distribution changes. I repeat, the distribution changes, not the absolute values themselves, because on average, the average methylation, if we combine all of these points here and here, the average stays approximately the same with maybe minor changes. Yeah, so the distribution changes and the peaks become lower, and in the middle appears another peak into which all of these huge peaks go, basically. And what it means, well, a physical intuition for it is that the entropy of DNA methylation increases, kind of like the randomness of their distribution increases over time. If we train some computational algorithm to see these patterns, then we can measure biological age of cells from their methylation patterns. And we can train these clocks on aging algorithms and apply them to the embryos, for example. It is a controversial method, I must say, because here we are clearly out of the training age range, because, well, embryos are not completely other organisms, basically, than either young people or adult people. But still, the theoretical background for such an idea is still in its infancy, unfortunately. But it's an interesting opportunity, nonetheless, to compare some age-related features of embryos which might provide us some additional knowledge. So what happens to methylation during embryogenesis? Well, a lot of things. Here you can see the timeline of how average methylation, which is laid on this vertical axis, changes over time during embryogenesis. Initially, a sperm cell and an oocyte are highly methylated, but upon fertilization, they become rapidly demethylated, and this blastocyst, during the blastocyst stage, the methylation is at its lowest. But after that, even more rapidly, methylation jumps back again during the gastrulation stage, and after the gastrulation, different cell types become differently methylated, I should say. So the average values are between 60 and 80% for different tissues. But the situation with progenitor germ cells, which will then again become the sperm cell or the oocyte, something very different happens to them, because their methylation drops again very rapidly and to the lowest levels across any other cell types for some reason again. And then all of these values jump back again to renew the cycle, to return to the initial levels. We could speculate that, for example, this first wave of demethylation or this rapid rise of remethylation or some other stages correspond to the lowest point of to the rejuvenation, to the process of rejuvenation. But these are all just guesses, because all of this roller coaster basically doesn't give us any clue about whether there is actually any methylation, any rejuvenation happening. Because, well, if the average levels of methylation don't give us any idea, any good idea about whether the cells are young at this point or whether they are older at this point. So let's stick to our idea of using DNA methylation clocks models. And it has been done by a group of Vadim Vladishev from Harvard Medical School. They took several data sets of embryogenesis and ran a number of different clock models on them. What they saw was that most of these clocks point at the period from embryonic days 6.5 to embryonic day 10.5 as the point of the lowest predicted age. But as you can see, the number of points here, the number of samples during that they can sample during this period is really, really low, as is usually the case with bulk samples, because there just isn't enough material in the early embryos to make a lot of samples from them. The way to go is to use single cell datasets, in which every sample is a cell and not just a bulk mass of cells. However, if we want to track epigenetic aging in single cells, we face a huge obstacle, which is the extreme sparsity of single cell DNA methylation data. This means that if you combine several cells, you might find basically no intersecting methylation sites. The coverage in single cells is so low that, the common methylation sites between many different cells may be even absent, which is not the case for bulk samples, because for bulk samples we've got quite huge coverage. And how can we make a machine learning model if our training features and our testing features such as in this case, if we train our clocks on, for example, cells like cell A and test them on cell B, we have nothing to test from. The machine learning model, the common machine learning model, will likely fail in this case. With all that in mind, an algorithm has recently been proposed, again by the Gladyshev team, to tackle this obstacle. And they have shown that, again, epigenetic age does decrease during the gastrulation phase of embryogenesis. In this case, they only took the single cells from the gastrulation phase. Unfortunately, we don't have other data sets after gastrulation, so we can see the decrease, but we don't see the increase after that. But still, the amount of cells is huge, and the significances are quite nice. But and yeah, by the way, this age decline, for some reason, relates really well with the increase of mean methylation, of average methylation. But when we look closely at this so-called SCH algorithm, which they use to predict all of this stuff, we can notice several flaws. Don't get too much scared by this slide. We will go slowly, step by step. Not too slowly, but kind of.
Evgeny, I should say about 5 minutes before 40 minutes interval rest.
5 minutes will be enough, yeah.
Okay.
So the framework consists of two main stages: construction of bulk reference and inference of age, well, prediction of age from this bulk reference. The general idea behind is that instead of how it is usually done, fitting one linear model across all the features, all the CPG sites, we instead train models like this for every CPG site, for every methylation site. And then, considering that in bulk we have a lot of sites and in single cells we have a few sites, we can still expect some intersection between them. And then we intersect these sites and make some, well, general inferences, basically on the language of machine learning. It's called maximum likelihood estimation, the solution of the maximum likelihood problem. And yeah, there are so many, well, there are three crucial problems to this algorithm I've mentioned here. We can talk about them a little later. But currently, what you should understand is that this algorithm is, first of all, really computationally slow and inaccurate in, well, many cases. Especially, we are worried that it is inaccurate in case of embryogenesis. So, how we improved this algorithm and what did we find about embryogenesis and machine learning in general by improving the algorithm and by using our new approach? This will be the story of our future meeting. Hopefully by that time we will already have published, well, at least have sent into print the articles about all this. And yeah, once again, I'm still researching the literature on all these topics. So my hypothesis and its arguments may change over time, but I hope today you've got the general idea of what happens when we are the youngest and how and why it may be important for the aging community, for the aging biology in general. Yeah, so thank you for listening. Please join our Journal Club chat for some future updates and stories. And I hope you've got some questions, which I'd be happy to answer. Thank you.
Thanks a lot, Evgeny. That was very high expertise talk.
Thank you.
So, okay, the 40 minutes discussion starting. Any questions from you about the topics covered, Evgeny? For example, maybe you're interested in embryogenesis. It's not a secret, I am involved in this research project too, but I know much less about the embryogenesis stuff. So I can't actually ask questions because I know all questions, because I know I know too much to ask questions to myself. So it would be very nice to hear questions from you, from the community, to understand what you think about this ground zero model.
I've got one. It's less so about the biology and more so about this kind of goal of creating a single cell methylation clock. Is this one that like a lot of different labs are kind of racing for now, or is this largely the domain of like the Gladyshev lab and like a few other kind of groups of researchers, or is this more of a like large scale effort that you see?
Well, as far as I know, since they published this paper in year 2021, since then there was only one paper that used this approach at all. They built the clocks on the blood samples, I suppose. They did some improvements to this algorithm as well, but the improvements were mainly they just compiled more data sets to train the algorithm on. But aside from that, I have not yet heard of any implementation of this algorithm besides what we do ourselves. So it seems like this is the tool that was needed for the field, but currently the field doesn't use it for some reason yet.
Dear Zhenya, thank you for your talk. And may I ask a couple of questions about the slides?
Yeah.
On the slide 11, there was a left graph about DNA methylation patterns drift towards mean with age. And I'm just wondering what the colors meant, like if, oh, you're showing, yeah, right. So this colors from, I think, violet to brown.
Well, it's just for the visualizational purposes. The violet are the ones that change negatively their methylation, the decrease in methylation, and the brown ones are the ones that increase. That's just, it means, well, different lines here are the different methylation sites. So for example, this site changed its methylation from on average like 1% to on average 50%. Yeah, and the other colors, well.
So different subsets of the DNA methylation sites.
Well, maybe subsets, maybe just the single sites. The authors don't state exactly what they show here because this is, well, just a schematic of sites.
It's a very strange one, actually. Like they've got the legends of the right graphs, and the left graph is like a bit mysterious.
It's empty, yeah.
Yeah. Imagine lines, they say. I don't know. Yeah, thank you for explaining. And the second question was actually about the extra embryogenic cells.
Yep.
And are there any works like you suppose that they are kind of behind the ground 0 point, or maybe higher than the cells come to ground zero? Like what do you expect their age is? And do you think that we can find some insights by looking at their probable methylation age? And do you think that any comparison with them is actually interesting or not? Thanks.
Yeah, thank you for the question. I think potentially we can extract some knowledge from them, yes. But there are several considerations. Well, first of all, yeah, I suspect that since evolutionarily they do not need to be rejuvenated for the embryo to be completely young, then they do not need they do not go after rejuvenation or during rejuvenation. So rejuvenation is allowed to continue after they are developed. Yes. So, but if we can actually think make up something about them is an interesting question, because, well, creating these cells itself is a very important topic, because in the developmental biology, for example, these cells are crucial to create, for example, artificial uterus to be able to incubate embryos for a long periods of time, because currently it's a problem. But how can we actually manipulate them, to look into them, to derive some conclusions from them? It's a very interesting topic, and well, it's just the trouble of data. Most of data and about extraembryonic tissues ends at this trophectoderm. And everything after that is usually just, well, at least in the epigenetic data sets, is usually just there is no data about it. But
so, because people don't look at it, or because it's harder to take it, or?
Well, I suppose it's just not deemed as necessary to look at them. But for example, when people collect such big data sets as this one, using transcriptomics, RNA sequencing data, they did include all of this stuff into their tree, which is kudos to them. Yeah. I hope to use all this data at some point in our research.
It's just interesting that you propose some dynamic that goes with the embryonic cells, and when you have kind of like control, which doesn't go anywhere, which shouldn't have any patterns, then yeah, it seems interesting to use it and to see what is there. What are the patterns there?
Idea. First of all, to check whether trophectoderm cells are older biologically than the epiblast cells, for example. That would already be some kind of a control. Yeah.
Thank you. Yeah.
Thank you.
I have a follow-up question to that. So, because the data you showed us, did that include trophectoderm layers? So that were they more methylated than the epiblast lineages?
Do you mean like here?
When you were showing like the single cell RNA data, I'm curious if they also looked at trophectoderm there.
At this point, the embryonic cells are just oh yeah, they did include, there are trophectoderm cells here as well, and they didn't even segregate all of these predictions into separate tissues. For example, trophectoderm, epiblast, and well, even mesoderm, ectoderm, and endoderm, they did include them. by applying RNA sequencing. Yeah.
Sorry, so you're saying they didn't segregate them, so we don't know the answer to the question if the trophectodome is indeed older.
As far as I remember, they sorry, I don't think I remember correctly if the trophectodome cells were in the data set already, because this is gastrulation and the trophectodome splits from all of the epiblast and other cells pretty earlier than that. And I suppose that they only included such things as ectoderm, mesoderm, endoderm, all of these embryonic layers only.
Got it.
This.
Do you know then another study where they did look at trophectodome and if that was actually older using transcriptomics?
Once again, as I said, this is the 2021 paper, and it is basically the only paper up to date which looked into the ages of single cell embryos, or even ages of, well, there is this paper of, sorry, yeah, this one. They published it in the same year by looking at the bulk embryos, and they did it for the single cell embryos in the same year. And these are the two papers that are the only papers currently which studied embryo, well, ages, which try to calculate ages in embryos at all. So, yeah. Unfortunately, I don't know anything about transcriptomic logs that were used to count ages, to predict ages in these datasets. But this is actually what we are going to do, so stay tuned for the updates.
Evgeny, concerning the previous question, could you remind me, is extra embryonic ectoderm is kind of trophectoderm, or it is another line? Extra embryonic ectoderm. Let's check, because actually the trap in his paper measured the age of extra embryonic ectoderm.
Oh, okay, that's great, because this is what develops from trophectoderm.
But yes, then concerning the previous question, the age of extra embryonic ectoderm is higher than age of 7.5 stage, for example.
So the trophectoderm is indeed the great control for rejuvenation.
I'm not sure, probably, but unfortunately we have only 1 point of extra embryonic ectoderm in the original paper, only from the 6.5 days after conception.
Strange.
Only 1 point, unfortunately, so we can't see any dynamics of rejuvenation or aging of ectoderm, extra embryonic ectoderm. But the fact is that the age is higher than in epiblast. Yeah, epiblast is a correct term here. Yeah, epiblast.
So we'll have to develop our transcriptomic logs to measure ages in all of these diverse tissues. Any other questions?
What were they using to come up with these lineage, like the end lineages? Was it just transcription factors or RNA expression?
Well, in the case of embryos, in the case of this paper here, they did use RNA sequencing, which was done in parallel with by sulfate sequencing with methylation sequencing. So yeah, we can use RNA transcriptomics to segregate them into different tissues. But it's not annotated in the original dataset.
Thank you. I'm just wanting to ask a re-clarification. You were mentioning about genome activation, or was it inactivation? If you don't mind clarifying what that is, and also totipotency, you were saying is a state of destabilization, and I was just asking to re-clarify why you think that is.
Sorry, can you repeat the second part of the question after the what is genome activation?
Oh, you said totipotency is a state of destabilization. So I'm just curious if you can re-clarify why you think that is.
Oh, okay. So the zygotic genome activation is what happens after. No, I should say it another way. After conception, when a sperm cell meets an oocyte, fuses with it, there is no transcription. The genome of the zygote is completely silent because it depends only solely on the factors from the oocyte, from the RNA that floats in the cytoplasm, from the proteins as well. But over time, slowly, as these proteins and RNAs get consumed, then the zygotic genome itself begins to open up, to transcribe its own RNA and to express its own proteins. And that happens in two separate waves. And yeah, by the end of it, it is completely, well, zygotic genome activation is what allows embryo to live off of its own material independently of what was inherited from the oocyte. And why I think totipotency is an unstable state, basically the major reason for me is that is this chromatin openness. That is, during the totipotent state, almost all of the genes can be transcribed, hypothetically, because there is no repression, almost no repression onto the genome. And we have the good genes, and we also have the bad genes. For example, these transposable elements. They are not completely bad because they appear to help during the embryogenesis. They appear to kick off some programs, but if they, and that is probably why the totipotent cell should be so much opened, because the transposable elements are required for the development. But as the longer this openness stays, the more side effects, I should say, these transposable elements will create. For example, they can disrupt the good genes, and that is a bad outcome. That's my major reason to think that prolonged totipotency is unstable.
Thank you.
Thank you. Oh, and also I haven't mentioned about another factor. This point here, ATL. ATL is alternative telomere lengthening. You could guess that telomeres are elongated during embryogenesis using the common telomerase pathway, but in fact, this is another pathway. It's alternative, and in adult organisms, for example, you and me, this pathway is so very rarely used, and it's mostly used by the cancer cells, in fact. But in development, in this early embryogenesis, when cells get cleaved, when the zygote proceeds through cleavage, this ATL, alternative pathway, does its job, and it only gives way to the telomerase pathway after blastulation. So during totipotency, again, this is not something somebody has checked like explicitly, but judging by the timing of telomerase beginning and by the timing of totipotency, the totipotent cell relies on this mechanism for telomere lengthening. And that mechanism, well, basically this mechanism is just homologous recombination of telomeres from neighboring chromosomes. And that is, again, not very stable, in my opinion. The telomerase pathway is more stable for long periods of time. I hope I've answered your question.
Evgeniy,
Definitely, that was really good.
I have a question. Do totipotent cells express lots of genes?
Oh, yeah.
Do they synthesize proteins?
Yeah.
Okay, so I know that sperm cells are the most highest in protein content. Sperm cells express a lot of proteins as an evolutionary selection. If there are some misfolded proteins, then these sperm cells die. So probably the same happens here. Some cells with mutations, which has misfolded proteins, they die. Is it confirmed that all of these proteins are actually expressed? Or is it just mRNA expression in totipotent cells?
Well, look, the trouble is that from the oocyte, the cells inherit a lot of different proteins. So the majority of proteins inside of the zygote comes actually from the oocyte, not from this sperm cell. But in sperm cell, yeah, the amount of proteins relative to its volume is big, but still the majority of them still comes from the oocyte. That is also the reason why in such experiments, when they take, for example, a somatic cell nucleus and transfer it into the oocyte, such a system, well, you've probably heard about these experiments of somatic cell nuclear transfer, such as this sheep, Dolly the sheep, you know. And such organisms are viable because oocyte appears to have enough rejuvenation factors, I should say, to counteract the accumulated age of the somatic cell nucleus. And yeah, that's why the proteins of oocyte are more important than the proteins from the sperm cell. Okay. And yeah.
Yeah, and I have a question about embryonic cells. So they're also totipotent, right?
No.
No, okay.
Yeah, this is an interesting question of whether embryonic stem cells are totipotent or pluripotent. And yeah, embryonic stem cells is what is well, by embryonic stem cells, the researchers usually mean something from these stages. So this is either the inner cell mass, which is this point here, or the epiblast lineages, this point here. They are already not totipotent, they are already pluripotent. And yep.
Okay, thank you. I have one more question. As I understand, the main problem of single cell methylation data is that it's very sparse, and most of the sites from these cells are not shared among cells. So wouldn't be single cell transcriptomics more reliable if it's less noisy?
In theory, it could be more reliable, yeah, thank you. But once again, we have the troubles of training and testing. because when we use one, well, in methylation, we don't really need any kinds of normalization or any other pre-processing steps. We can just use the raw methylation values as they are. Well, almost. But in case of transcriptomics, we have two data sets. One, the training one, and the testing one. Well, the embryonics. And not even two data sets, but we should better have three of them: the training, the validation, and the testing. And all of them must be preprocessed in some way that these relationships remain the same. And how can we do that? Well, that's a very deep question on which we are still working. Yeah.
Thank you.
But I want to add here that in principle, if we measure the transcriptomic age with a kind of transcriptomic clocks, in reprogramming dataset, for example, especially in the single cell reprogramming dataset, we actually see this decreasing pattern of age. So I think, and some of my colleagues even tested this, that single cell transcriptomics shows this ground zero-like pattern too. However, to my best knowledge, this result wasn't published. I don't know why, but we didn't reproduce this result with transcriptomic logs on embryogenesis dataset, but we reproduced it with reprogramming dataset.
Okay, thank you. And one more question, Zhenya, how does a data set of your dream looks like? I mean, single cell methylation.
Well, basically, it's something like this. Because for transcriptomics, we've got such a great data set, or even better, something like this. But it starts here on day 9.5. The ideal one would start at conception, just after conception, and ideally, again, I'd like to include the germ cells into this dataset. So everything I've shown here, basically, with my red arc, should be included into this dataset, starting from the development of sperm and oocyte, and ending with the birth.
Yeah. With
temporal resolution of at least 1 day. That would be the perfect dataset, I suppose.
How many cells per sample do you want?
Well, I'm not sure about the exact values, but well, for example, we've got this dataset of embryogenesis that I've shown, and there were like 250 cells per stage. That would be maybe overkill, maybe just enough. Well, for me, at least 100 cells per stage would be great.
Okay, thank you.
Are you trying to make a dataset for me?
Yeah, sure. I'm going to steal it from AltaSlab.
Nice. Tell me how it goes.
Okay.
Evgeny, by the way, one question which actually, how do you think why aging clocks are trained on bulk samples and then they are applied to single cell samples? Is it correct? Do we can be introduce bias to bulk samples, to bulk clocks by tissue content bias, for example, or something?
In principle, yeah, of course we could. And we probably do introduce some bias, but what else can we have? What else is left for us? I mean, for this, we don't have an option to train on single cells and to test on single cells, at least for DNA methylation. So basically, we should validate. It looks to me that we should validate both on bulk tissues and on single cells outposts, and to get the combined idea of what is actually of whether we have any biases between them. But that would require such a data set in which we would have both the bulk samples and the single cell samples of the same tissue, for example, and that is really difficult to obtain.
Okay, 5 minutes left before for the end of today's session. Yes, you scroll in the right side. Yeah.
Just the slide of
not this. Please, could you show the this slide? This is very cool slide. I surprised that you didn't include this in the presentation.
Well, it's a long slide to talk about, so
yeah, because in reprogramming, even in the bulk samples, we actually see this erasing of epigenetic age and achieving 0 epigenetic age, which is also treated as biological age. So it is actually something what we maybe this one of not many evidence we could use to validate ourselves in the embryogenesis story.